Good morning, everyone. I am Tamas, Shisha. It's an absolute pleasure to be back to Paris. I am from Novartis, translational medicine immunology, and I was asked to just give a short introduction to this session. And it's always amazing for me to come back to Paris. We've lived here with my family. Actually, my two daughters were born in Saint-Cloud, so part of my heart belongs here, even though we left 11 years ago. In this next session, we will talk about next generation of gene therapies, and this session specifically is going to be about how the use of CAR-T can go beyond the cancer therapy, how we can improve the delivery to solid tumors and applications beyond cancer. This introduction will be followed by short talks and a roundtable discussion. And my introduction is going to be about our own Novartis experience of going beyond cancer. And I'm going to present to you the Sentinel data from the first 3 subjects of this ongoing clinical trial in severe refractory lupus nephritis treated with CD19 CAR-T therapy. So the whole story really began with the cases presented by the group of Professor Schett in Erlangen. Many people have been thinking, obviously, about going beyond cancer with CAR-T therapy, but it was really we in Novartis have been thinking about it, but there were many pros and cons, many risks, and doing the first step was an important one, and Professor Schett indeed made this first step, and the data was really quite spectacular, as you can see on this slide, with a dramatic improvement of the disease activity score, SLEDAI, on the left, complete depletion of the B cells, and not just peripheral B cells, but most likely, obviously, the deep B cell depletion in the tissues. Also, complete depletion of double-stranded DNA and interferon, while maintaining, as you see, limited impact on protective antibodies. So this was, so to speak, the trigger for us to also go on this endeavor and set up the study. So this is just a background slide, but I think I really summarized the contents already. So in terms of the methods, so this is an open label, a single-arm, multicenter study. Multicenter, we also have centers in France, several, one here in Paris in La Piquier, to assess safety, efficacy, and cellular kinetics, so YTB, in patients with severe refractory lupus. So I'm going to present this cohort of patients, the Sentinel cohort, that was dosed with 12.5 million cells. And we looked at CAR-T kinetics by qPCR and flow cytometry, and we looked at a number of other secondary endpoints, as you will see on the next slides. So here is the baseline disease characteristics of these first 3 Sentinel subjects. So you see that the patients were between 38 and 50 years, male, female. All of the patients received the same lymphodepleting chemotherapy before receiving YTB. They had a long disease duration of more than 10 years. All of the patients had high disease activity. You see the SLEDAI scores, 22, 14, and 12. And all of the patients had several major organ involvements. This is the safety profile at the time of the data cutoff for this Sentinel review. So you see all the adverse events. So this is patient 1, patient 2, and patient 3. In color, we have the grade 3 and the grade 4 events. So these are obviously the more severe ones. And as you can see, these are adverse events that one can really expect after the lymphodepleting chemotherapy. So these are all cytopenias, lymphopenia, neutropenia, leukopenia, lymphopenia. So these are reported in clinical trials as they should be, but of course it was nothing that we did not expect, and the data monitoring committee had really no concerns, and neither did we. So on this slide you see the depletion of CD3 T cells with a pretty quick recovery. So this is the result, of course, of the lymphodepletion pre-treatment. And here the CD19 cells which remain depleted throughout the period of time. So the longest follow-up was patient 1 until day 91, clearly showing a distinct effect of the CAR-T therapy as compared to just the effect of the lymphodepleting pre-treatment. Here you see the efficacy. So SLEDAI improvement going from baseline values. So this line is day 1, so when patients received YTB, going down quite dramatically. So this patient 1 was at SLEDAI 0 actually at month 3. This is the physician's global assessment of disease activity, also showing remarkable improvements. And here you see double-stranded DNA and complement C3 and C4 improvements. We also plotted the efficacy improvement on top of the data that was reported by Professor Schett's group in Erlangen. So the gray lines are the data from Erlangen, and the colored lines are the patients ongoing in our own study. We just wanted to see whether the effect and the effect size is in line with what we would expect based on the Erlangen data, and we were comforted to see that it's very much in line. And so this preliminary data really prompted us to go on and to continue the study. And currently we already have treated 10 patients, so since this data cutoff, so it's going fast and more data is to come, and we will be proud to share the data with you. Next opportunity will be in Bruges, in Belgium, in March at the European Lupus Conference, and then in June at the EULAR, we also plan to present an update from this study. And with that, I would like to thank you for your attention.
Thank you very much for this great introduction.